Hi everyone! Today we're going to learn about transcription, which is the first step in protein synthesis. Transcription is how we create a single strand of RNA from DNA, and its purpose is to carry information from DNA out into the cell. DNA cannot leave the nucleus because it's double-stranded, so it's larger, but also because it's the most important part of the cell. The cell could not function without it, so it stays inside of the nucleus to be protected. RNA stands for ribonucleic acid. It is made of nucleotides as well. So we have phosphate, sugar, and nitrogen base, but there are a few differences. So the sugar in RNA is ribose, whereas in DNA it's deoxyribose, which is where the D comes from. For nitrogen bases, 3 of them are the same, but instead of T, thymine, we have U, uracil, so that gets replaced. The process of transcription starts out by the DNA molecule being unzipped, but it's only going to use small sections, which are called genes. Then free RNA nucleotides are going to be added to one strand of DNA. So during replication, we were adding bases to both sides, but in this case, we're only going to use one side and one section. So those RNA nucleotides will base pair to the DNA. Then mRNA, M stands for messenger, is going to leave the nucleus and carry those instructions out into the cell or into the cytoplasm. The DNA then reforms the double helix structure by coiling back together. So this is a basic picture of transcription. So remember, we are in the nucleus because that is where DNA is stored. The DNA is going to unravel, unzip. Those hydrogen bonds are broken. RNA nucleotides are going to be added to the DNA template strand. And then when it's done, when it has synthesized all of the RNA that it needs for that gene, that mRNA is going to leave the nucleus through the nuclear pores. So here's a quick video showing you transcription.
Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Okay, so you do not need to know any of those specific proteins. You just need to keep in mind that we're using one strand of DNA. I hope you noticed that, as I mentioned before, it's only copying from a certain section of the DNA, and we're adding RNA nucleotides, and then that will leave the nucleus. Okay, and then the last thing is just a little bit of base pairing practice. So You see on the left side, we are doing complementary DNA nucleotides. So A is going to bond with T, C bonds with G, just like we've been learning. But with RNA, when you're going DNA to RNA, A is going to bond with U because there's no more T. So if you look on the right side in the all black DNA to DNA, our sequence would be A, G, C, A, T. But on the bottom, when we're making RNA, we would have U, C, G, A, U, A. So summarize your notes. Make sure you use the terms RNA and transcription, and then write any questions that you have.